So this article is pretty cool. It comes from Wageningen University and Research, and it's about a new automated plant phenotyping device. And that probably sounds really interesting to a lot of people. Well, a small group of people, particularly people who I know. But anyways, I wanted to talk about it because I thought it was pretty cool. So, this new phenotyping device at Wageningen. If people want to find the article, you can look up Wageningen new phenotyping device, and it should come up for you on any search device that you use or search service. The first paragraph is probably the most informational, so I'm just going to read it verbatim. It says the Shared Research Facilities, the SRF, of Wageningen University and Research, the WUR, has recently purchased a new mobile device suitable to automatically phenotype plants. The company Photon Systems Instruments, PSI, from the Czech Republic has developed the Robin PSI Plant Screen TM system based on specific requirements set by Wageningen University Research. It has been installed in WUR's location, Unifarm, and it is the first device that has arrived as part of the new Netherlands Plant Ecophenotyping Center, or the NPEC, which I've talked about previously. For people who don't know, there's a saying that goes, and for horticulture I've been exposed to it quite a bit. They say, if you ain't Dutch, you ain't much. And I don't necessarily agree with that, but I do have to agree that the Wageningen University is phenomenal. If you ever get a chance to go to the Netherlands, if you ever get a chance to go take a look at some of the interesting research that they're doing and the technology that they have access to, I think it would really help you out. No matter who you are, honestly. Even if you're at the top of the horticulture or agriculture game, there's something always coming out in Wageningen. And they didn't pay me to say that. They're just that cool. Anyways, so they have this machine, and I'll link to this in my YouTube video, which this is going to go on YouTube for those who aren't aware. I always like to mention it. And if you have any questions, please leave them in the comments. But I'm going to put a link to this in the description so you can take a look at the same article. It's from Wageningen, and they're likely to have more information if you want to contact them. It even says here on the article, if you have questions about the device, you can ask Rick van der Zette. Hopefully I'm not destroying that name there, but I'm pretty sure that's how you pronounce it. Anywho, if I go on... Oh yeah, thanks. This is from Mood Made Farms. Those who are aware of them, they're from Southern Humboldt. They're really cool people. I really enjoyed working with them. Definitely recommend them. But to continue, it says here that the Robin PSI plant screen system is a standalone device that can be used to digitally phenotype individual plants in pots up to 50 centimeter plant height, or trays with maximum of 20 small plants. For instance, Arabidopsis, which is Thale cress, which is commonly used in scientific research, so that would be very helpful for automating that process. The Robin PSI can be easily transferred to a climate room or a greenhouse compartment to accommodate easy access from running experiments. The Robin PSI is equipped with 3 sensors, and these are pretty cool. A chlorophyll fluorescence imaging unit for dark adapted photosystem 2 PS2 efficiency measurements, a red-green-blue imaging unit with top view and multiple side view options, and a 3D laser triangulation scanning unit for 3D reconstruction images. This thing can take the plant and reconstruct it in 3D. That's pretty amazing to me. And that's the kind of Star Wars stuff that I'm expecting from the rest of the world, hopefully. When I say that the Dutch or Holland or the Netherlands, that they're very serious about their agriculture and horticulture, this is the kind of stuff I'm talking about. And if this is actually more commonplace and you happen to know better, please send me a message and link it to me, because I'm pretty sure this is the only place where that exists, which is, again, I've said it already, but it's amazing to me. They say it's easy to use. I wonder about that. But if it is, that's also a really cool benefit, of course. Um, right down here, they say that under the title characteristics, they say depending on which sensors you use and the size of the plants, a measurement will take a few to several minutes per plant. Pretty fast. If you want to use the CF unit, you will need to take into account that a dark adaptation period is needed, which takes 5 to 20 minutes per plant. Again, not that long at all. It says that the Robin PSI, and here's where it gets really cool, the Robin PSI is able to collect the following physiological and morphological data. Max PSI2 efficiency, biomass. Let me put that again. The following physiological and morphological data is able to be collected by the Robin PSI. Max PSI2 efficiency, biomass, volume, project leaf area per individual leaf, main stem length, plant height, number of leaves, internode lengths, color, etc. Upon capturing the images, it stores the raw data, segmented and 3D point cloud, and all the CF and RGB images for further analysis by other tools. So you can take all of this information and you can even export it to another system, which is great. That's really, really cool. So, and then they have a caption for a picture. This article is not large. I definitely recommend people to just read it over. It's literally 1, 2, 3, 4, 5, 6, it's like 7 paragraphs. It's not very complex. I've already gone through half of it at this point. But it says here that under a picture caption, one of the research topics suitable for this technology is to study the influence of environment on the phenotype of the genotypes that are tested, P equals G plus E. And for small plants, G times E can be investigated. If plants are exposed to different biotic or abiotic environmental conditions like salt, drought, heat, or pathogen or herbivore stresses, then you can find more information about that reaction. That's pretty cool. Pretty cool. And this is just one of several systems that the NPEC is coming out with. And for those who are just joining and wondering what I'm talking about a phenotyping device at the university in Wageningen. And it's part of a center from the Netherlands called the Netherlands Plant Ecophenotyping Center. And I know that they have other systems that are meant to basically study the phenotypic expression of plants under different conditions in order to research better cultivars moving forward, depending on what kind of environments they're going to be found in. So, for example, it's pretty obvious when you really think about it, what they're trying to do. They're trying to take plants and make them better for a hotter environment. Make them more salt tolerant in an environment where that's very common, where saline water is very, very common. I know that I talked with a friend of mine who was picking my brain regarding a way to bioremediate, where was it? It was some sort of salt flat or something. I think it's in California actually, where the rivers have just been dammed up or they've been diverted, like the Colorado River, for example, and it's just barren and salty. And they were thinking, hey, what if we took the there's a grass, and I think it is actually called marsh grass or salt grass. And I don't actually remember the name off the top of my head, but they were thinking, why don't we just take this plant, which is incredibly salt tolerant and actually bioaccumulates salt as a defense mechanism, and plant it all around these salty flats and sort of take out the salt while you're growing the plant, which is kind of cool. And so this sort of reminds me of that. You could perhaps find a cultivar or help grow a cultivar of plant that could be even better used and utilized for this sort of a thing, both in the place my friend was talking about and elsewhere, where there is quite a bit of salt, either because of pollution or because of just the natural environment growing out that way. And if you want to make the area a little bit more arable, that might be one way that you can do it. So anyways, that's kind of the main reason that I brought up this article because I thought it was such an interesting topic for discussion, or at least me dictating to people about it in my enthusiastic fervor. I don't want to read the entire article to you guys, but basically, if you have any questions or comments, or if this sort of stimulates something in your head, please talk about it if you are so inclined. While you do that, I will read this paragraph. It says, the NPEC, which is the Netherlands Plant Ecophenotyping Center, is a national facility we are developing, approved and funded by the NWO. This means that the Robin PSI is open to academic users as well as industrial partners, upon consultation with Rick van de Zedde. I hope I said that right again. To encourage usage, this device is available at no costs. And no costs will be charged until the end of this year. So if you're going to the Netherlands, you should definitely hang out with the Wageningen University and take a look at it for sure, for those of you who would be interested in such a thing. I'm definitely getting people joining. Hello, I appreciate you guys coming in and talking about this really odd phenotyping device. Odd and amazing. It's called the... What was it called? The Robin PSI Plant Screen TM system. And it's by Wageningen, which is in the Netherlands, like I just mentioned. And it's built by the Photon Systems Instruments Group in the Czech Republic. So a lot of trans-European collaboration coming together, which I think is pretty cool. And yeah, that's really all I had to talk about. They say of victory, you should know when to stop, and don't continue on, and you might go too thin. So I'm not necessarily going to leave it here, but I might, because I don't want to ramble and take up so many people's time. And also my time. Putting these videos out on YouTube is kind of arduous sometimes. It's a long time period. The last one I put out was I had to break it up into 3 parts because YouTube didn't want me putting out what would essentially be a feature-length film of The Hobbit of just me talking to the camera. Most people don't have time for that. For those who do, though, I really appreciate it, and I hope that you guys take something away from it. If I just click on the Wageningen University page, there's a lot of biotech going on. Very cool. Very cool. It's weird because most people I know don't even talk about this sort of stuff. Either that's because I'm in a niche environment myself, or it's because in general people are just not aware of what Wageningen does. But here we have a TED Talk by Wageningen University, and we have a symposium and all sorts of stuff featured here. Theory of says, get the sword out. This sword, get this sword out right here. This one right here, the sword. You're probably curious about that sword too. It's really kind of got a funny story to it. There's nothing about that sword that's battle ready at all, but it is kind of nice. The reason why I have it up here partly is because I thought it looked kind of cool, and partly because it's kind of a heartfelt present. Since I'm not actually going to get it out for you, I'm going to talk to you about it instead. Maybe in another video I will, because it's attached to the wall in a way that would make it very difficult to get it out. So, not because I don't want to necessarily whip it out. Yeah, I'll whip it out later. But it's a birthday present that I got when I turned 21, actually, from a group of close friends. So I thought that was kind of cool. Yeah, OG Higher Being says, that sword is his main IPM tool. Well, only in one-on-one combat, as is honorable. Although, when it comes to russet mites and aphids, I don't tend to practice honorable combat. Anything goes, because that's how they are, too. They're always invading, so, Actually, I think that would make a really cool video game, I think. It's if somebody took, I've been thinking about this and ruminating on it for a while, actually, because if you had, if you got the story and the design right, you could make a pretty fun game of, people living in on, what is essentially a large plant, and, fighting like pests and things, like a small scale like IPM soldier or something. Most people wouldn't really. You'd have to market it right, but I could see that being kind of fun for people who are like interested in that kind of stuff. Work on your sword mounting skills. Oh well, it's really not not something I want to spend too much time on. Maybe you can teach me. But yeah, I mean, I'm live at this point, but for those of you joining in, I was talking about this really cool phenotyping device in the Netherlands called the Wageningen University and Research. I would play the hell out of that. Yeah, Theory, I would too. Wouldn't you? Like, if you, like... I'm glad you guys are saying this. I'll talk about this then instead. Because I was thinking about it, like, in sort of a, like, magical realism sort of way, I suppose. Yeah, like, you could have, like, the people who live on the plant, and, like, it doesn't even have to be, like, in your face about what the setting is. It just, like... This is the plant, and it's like microscopic. So you got trichome forests, you got glandular trichome, like bulbous trees, essentially, and you've got these giant capitate resinous glands and striculate trichome hairs, and all that kind of stuff would be pretty cool. You'd have to navigate through that, right? And maybe you'd have, like, if it was like an RPG, if it was like a role-playing game, I suppose you could have, like, different classes, right? So maybe in a basic sort of way, you could have, like, a warrior who maybe uses, like, a sword or, like, shears or something, right? Make it cool, don't make it, like, lame. Or maybe make it lame. Maybe people would like that if it's kind of like he's got, like, half of a shear, just like one half of it as like a blade. Or you could have somebody who's like a magic caster, but not, instead of using magic, they like use like biocontrol products or something. Like they use Beauveria bassiana spores and they like throw them at them or something. Might make me play video games again. Will you be releasing it for the N64? Oh, I don't know. It might actually be easier to just do this on the N64 or an emulator, or do a ROM hack or something like that. For those who don't know, I was ROM hacking a video game to do something kind of like this, but not quite. I don't have the skills nor the programming knowledge in order to do it. But I was trying to create a sort of a medieval fantasy video game about 3 years ago. I also worked with the Pokemon Generation Zero guys, if anyone's familiar with those people as well. But now we changed direction quite a bit ago because Nintendo was getting very litigious with their intellectual property, and we didn't want to be sued over it, so we dropped it, even though it was a pretty cool system. Anyways. Oh yeah, or like a beetle mandible. Like, yeah, like totally having like a, like maybe like beetle armor. Like have a stag beetle, one of my favorite kinds of beetles, just have that like as their armor. And like, or maybe like a fighter that doesn't use weapons. They just have like the beetle horns and they can like grab people and like tussle with them or like bear them down or something. I don't know. You could have like a magic caster, a fighter. What else could you have? You could have like a rogue. I don't know. You could have like a dark magic user who is somebody who uses like avamectin or something, maybe. Or you could have like, or maybe don't even get political about that. That might be a bad move. Like some myclobutanil dark magic. Maybe that could be the central like theme of the game. That like some people use these chemicals and it can have like a very Aaron Brockovich sort of like environmentalism theme where like there's a kingdom and they utilize the chemical agents, but they're really dark magic and they're just killing the environment because the local biocontrols, which could be another sort of class, maybe like a tamer who uses like Persimmilus and Californicus and Phalanxus and that sort of a thing, and maybe summons them. That would be kind of cool. You could fight like aphids and shit. Yeah, or like russet mites or any kind of thing. Like Yahomi would be a ladybug or something. Yeah, like, or you could summon them, or they could actually be your companions, or maybe there's like cities and towns full of them. As you can see, I've thought about this quite a bit, and it really sucks that I don't have access to like the resources necessarily to like materialize it. But as things change, indie games become easier and easier for people to make and collaborate with people about. I think it'd be very important for it to not be like, I mean, when I think of things like that, just like my own channel, I'm not trying to like get super famous or sell a bunch of copies or anything like that. That's not really the intent. The intent is to make something that I think would be a really cool thing for people who would be interested in, and then do that. And I guess I feel like it would be possible. I feel like it could be popular for not a whole lot of people maybe, but for a lot of people if it was done right. How about an anti-hero with some Eagle 20 potion? Yeah, like that's what I'm saying, right? Like that could be the theme, is like maybe they're producing really good something or plants or whatever, or just their existence is fundamented on this sort of like dark secret that they're using this chemical compound. Well, dude, I gotta bounce, but I want to hear more about this. Okay, well, I'm glad that my like weeaboo anime video game ramblings are entertaining to people. Pot farm or just saying, it would be fun insider type game. Yeah, right. Like if you marketed and branded it to like the cannabis community or like the horticulture community. The part of the problem with that demography is that, like I like to joke with people, if I wanted to be really famous, I wouldn't have decided to go into agriculture. Why? Because as I'm told by like every single person in the agriculture industry that's like over 30 years old, and cards on the table, I'm 26. So for all those people, they're always telling me about how like we need new blood, new people aren't coming in, and it's like, yeah, because it's not attractive to be a farmer. It's not attractive to be a horticulturalist. The concept, the idea is attractive to a lot of people, including myself, which is why I did it. But it's not like people aren't running around trying to like do a whole lot of manual labor, for example, and a lot of people are treated pretty poorly in a lot of industrial farm agriculture settings. So it doesn't make people want to like get in the industry, and it doesn't pay very well too. And it's the economics of like horticulture for a lot of places is lacking, for a better word. But you're right. Like I could see that sort of demography of like younger people who like grew up playing Pokemon or something like that, which is another thing I've thought of. You could have like biocontrol agents that you go out and catch and then maybe level up and then utilize on other bad organisms, and maybe you can like catch a russet mite or like catch an aphid essentially, or catch a persimmilus mite or something like that. You might have to change the names up a little bit or something, but yeah, I could definitely see that being something people would like. It would be Top Grade Medical says, what were you saying about the Netherlands? Yeah, good question. Let's stay on topic, Matt. What I was saying about the Netherlands, for those interested, there's a new automated plant phenotyping device by Wageningen University. Well, it's not by them, it's at their university. And Wageningen University is a really cool place. I've never been, but I know that they do some really interesting research, and if anyone has the opportunity to go, I would highly encourage them to. And if you missed anything that I said, you can find this video on my YouTube channel, for those who aren't aware, Zenthanol, Zenthanol. And I can read this paragraph for you just to get you started. It's a very this article will be in the description, so you can read it yourself. But essentially, they say the shared research facilities of Wageningen University and Research has recently purchased a new mobile device suitable to automatically phenotype plants. The company Photon Systems Instruments from the Czech Republic has developed the Robin PSI Plant Screen TM system based on specific requirements set by WUR. It has been installed at Wageningen University's location, Unifarm, and it is the first device that has arrived as part of the new Netherlands Plant Ecophenotyping Center, the NPEC. And what all of that means is that they've got this cool little device that can measure things and take that data and create a 3D image, and it can measure things like chlorophyll fluorescence. And down here they talk about more things that it can, some of the data that it talks about being able to record are like the, here is it, where does it go? Where is it? So the photosystem 2 efficiency, biomass, volume, project leaf area per individual leaf, main stem length, plant height, number of leaves, internode lengths, color, and more. All of this data can be taken and it can be analyzed and also exported to other systems, and then they can use this information for breeding. They can use this information to assess physiological reactions to certain stressors, so you can breed cultivars for better environments or better or more efficient usage in different environments like heat, hotter places, saltier places, colder places. And as climate change happens, and is probably going to happen quite a bit if we don't get our act together, this will be pretty important stuff, because climate change is affecting and has always affected, well, because the climate is always changing a little bit, your climb definitely changes how you approach things. You would not approach things in a desert the same way you would approach in another kind of environment, right? All based on imaging? That's a good question. Second ton 420. Yeah, I guess it's not just pictures, because that wouldn't really make sense, right? I guess it does other sorts of things too. It probably uses like lasers and other sorts of things to get readings on some of the like fluorescence and sorts of things like that. I don't actually know all the details. But yeah, isn't that pretty cool? Yeah, holy coolness, right? Resin Lung says, the other day I noticed a comment you made in Miami Mango's post that his issues could be due to agrobacterium, and that even if his issues were not, that you had seen this in Cannabis Like Freak Show. Yeah, good point. I did say that. That was regarding they had what kind of looked like a crown gall. And if you're not familiar, and I'll have a picture of this on YouTube at this point right here for you guys to be able to see what it looks like. A crown gall in cannabis and other plants looks like this giant tumorous growth, and it's caused by an organism called agrobacterium. And anyways, are you saying that is the cause of the freak show? Yeah, no, it's not necessarily the cause of the freak show. It's just that the agrobacterium causes hormonal changes in the plant. That's how it's able to create the tumor. And it is possible that it could also cause other sorts of hormonal issues. When I say issues, I really mean to say differences and changes in the host plant. So it's not outside their own possibility, because it's happened in other plants where agrobacterium can cause the plant to dud. Like in roses, for example, you'll have stems that would normally turn into flowers that you would cut, but instead of turning into flowers, they just turn into what are called blinds, which is what dudding is in cannabis speak. It's when the main stem or different stems, they terminate into foliage and no flowers. And that can happen for agrobacterium. That can happen with phytoplasmas, which I think could be a possible reason why some cultivars of cannabis and other plants, I mean, it's known to be the case in other plants, but it could be a possibility because there are hemp phytoplasma, which are a special kind of bacteria, that could be causing other sorts of hormonal imbalances that could change the way that the plant grows in a special way. And the Freak Show cultivar definitely has and had a similar sort of development that Miami Mango's plant did. So I was just kind of relating that they look similar, not that they are necessarily caused by the same sort of thing. It's also possible that different pathogens or different stressors can cause the same thing, or what looks to be the same thing, in two totally different plants, or in the same plant and they are totally different causative agents. That's also possible. Yeah, so you're very welcome for that really long-winded answer. Hopefully it was helpful and not super confusing. Second ton 420. When you say this is such a cool concept, do you mean the... Oh yeah, you're talking about the Netherlands thing. Yeah, isn't it a pretty cool thing? I can't speak more highly about them, and I've never been to the facility, so like, I guess I can't talk from personal experience, but I can only talk about from, my experience of like working with people who are from the university or who've worked at the university, and they're always crackerjack people. And so that's just been my experience. But again, not personally and not physically at the location or anything like that. But they're always innovating on horticulture and agriculture work, so I'm always finding new information regarding lots of things in agriculture, and I definitely recommend people to like take a look at their like social media. They're pretty good about their social media and getting information out too, even though the agricultural environment and the agricultural sort of social media spheres kind of ho-hum, I suppose. I was late, still speaking on the Pheno system. Yeah, the long-winded answers are best. You, sir, are a wealth of knowledge. Well, I appreciate that. Aw, shucks. It's kind of nice to hear that. Definitely, I've been harangued before for kind of talking over and over and over again. But I think the problem is not necessarily with me, always. It's sometimes it is my fault for finding the wrong demographic to talk to. On Instagram, I can just talk about whatever I'd like to, and here I am. And people who like it will stay and follow, and people who don't will not. So maybe the problem is with me. It's just talking to the wrong people about that subject, which I know a lot of people have done for other sorts of subjects, so I'm probably in that boat too. Yeah, so I really appreciate the compliments, and I appreciate that people are getting something out of it. Definitely. Yeah, you're very welcome for the information. Have a good one. I appreciate you, Keebler. I appreciate you. Yeah, so I guess, maybe I'll stop it here, perhaps. Oh, but Space Cowboy just joined, so he's probably wondering, or they're probably wondering what's going on. Try not to gender people. You never know who you're talking to on the internet, so. Anyways, yeah, so I was talking about this. So this whole video, if you've just joined and found out that unfortunately I'll be signing off pretty soon, disappointingly, I will be putting this up at my YouTube channel, Zenthanol, Zenthanol. If you haven't subscribed, I don't want to be that kind of person who's like, smash that like button, smash that subscribe button to join, and blah, blah, blah. But like, if you want to get this information, that's how you can, find it. Like, it's just a logistical question of whether you want that info or not. I also have a Patreon for people who want to, chip in monthly, and I have a PayPal for people who want to chip in singularly or whenever they want to, I suppose, and not at a regular level. But this is the sort of topic of conversation I like to do, and I just became aware of this sort of phenotyping device. So if you're interested in this really cool, fancy schmancy phenotyping device at Wageningen, take a look at the video on my YouTube channel, which it should be up. The video, that is, this video right here, should be up maybe tomorrow or something like that, or maybe tonight. I'll probably start uploading it tonight. So I have a little update for what I was talking about earlier about the Wageningen University. And what that update is a post that I made earlier in the year. And if I check on Instagram, the post is from April 13th, 2018. And in it, I'm just going to say what I said verbatim, but it has to do with the system that I was referring to, the phenotyping system, the device. And I say, the Dutch Minister of Education, Culture and Sciences recently announced the funding of the Netherlands Plant Ecophenotyping Center, the NPEC I was talking about, as part of the National Roadmap for Large-Scale Scientific Infrastructure of the Dutch government. The NPEC facility is an initiative of Wageningen University and Research and Utrecht University and cost 22 million euros. The facility will study plant performance in relation to relevant biotic and abiotic factors across a range of scales, from molecule to crop, nanometer to kilometer, using digital phenotyping systems. In the precision mesocosm level ecotron, or ECO, or ECO, plant-plant and plant-microbe interactions both above and below ground will be studied. At a level of centimeter to meter, the plant-microbe interactions phenotyping module, or PMI, will be used for high-throughput research on plant-microbe interactions, from the molecular level up to the organelle level. The Multi-Environment Climate Chamber module, the ME, has been designed to study the molecular basis of plant responses to multiple environmental factors. The High-Throughput Phenotyping Climate Chamber module, the HTP, allows for automated high-throughput screening of plant genotype under highly controlled environmental conditions in the greenhouse phenotyping model, or the GH model. Still more. Integrated whole plant phenotyping will be carried out in testing numerous crops, from seedling to harvest. The Open Field Phenotyping module, or the OP, provides an outdoor mobile drone and vehicle-based phenotyping system that can study individual plants in small plots or large fields, which is going to be pretty useful. It's hard to overstate how technically brilliant this setup is ostensibly, those are my words. I'd like to see similar systems in other parts of the world if they prove even half as valuable as they purport. And yeah, only I am the sort of person to use the word purport in a sentence that I know of. So those are definitely my words. Right, so in April I was talking about this earlier, this NPEC system, but there's a ton of other things too, and if you're just joining and you didn't get that entire diatribe, you can check it out on YouTube. But essentially, I wanted to do a little sequel. Two things. One, I wanted to say a little bit more about the NPEC and Wageningen and the sorts of technologies that they're investigating in, and they're investigating, like this 22 million euro research facility, this combination of Wageningen and Utrecht University put together. I think it's really awesome. And I also want to talk about something that's not related at all to any of those things, but I'm going to put it in the same video because often I have one or two pictures or one or two topics I like to talk about. And the second thing I like to talk about is just sort of an interesting, I guess you could say it's like a lifestyle thing with plants, or rather how they make their food. I was watching a video recently in which somebody was making the point that brambles, or at least a particular species of bramble that they weren't necessarily they didn't really mention what species it was, but it had really hooked thorns. And they talked about how this could be considered carnivory in plants. And I immediately knew where they were going with this. And I thought it was really an interesting thing to talk about, so I'm talking about it. So what is that? So essentially, there's this concept in carnivory and omnivory and herbivory called paracarnivory. And just like a paramedic isn't necessarily a medical person, a paracarnivorous plant isn't totally carnivorous, but definitely makes it gets some sort of benefit from animals in a more direct way than having other organisms sort of churn through and process their bodies and and bioturbate the nutrients in the soil or whatever substrate they're growing on. So what does that look like? Well, for the bramble, it works like this. If you're a farmer and you happen to have livestock like lambs or sheep, are those the same thing? I forget, actually. But, you got some sort of a creature that's very woolen. and it's walking along the fence or whatever, and it gets stuck by some bramble, and maybe like there's like almost like a noose sort of like structure to the binds, and the wool gets stuck in the hooked thorns. And so what'll happen is that it will just kind of die. It'll just die that way. It kind of gives up trying to like push against the thorns. And another animal will come around and eat it, and it will either kill it right there and eat it on the spot, or it will die of pneumonia maybe, which they mentioned in the video. And like a crow will come out and kill it, or some other organism will come out and kill the animal, or it will just die there. However it dies, that doesn't matter. But what does matter is that as long as the body is left there, in whole or in part, honestly, it doesn't really matter, because anything that's more than 0 is a benefit to the plant. Because for obvious reasons, the bones and meat and all of the different parts of its body it'll rot, and it will, die and die. It's already dead at that point. And it will just get into the soil, and it will essentially fertilize the bramble, simply for getting close to it and getting stuck into it. And honestly, I think that is somewhat qualifying it for what's called paracarnivory, which is like not direct carnivory like a Venus flytrap or a pitcher plant or a sundew or something like that, which most people have at least seen in like Home Depot or something, where people are buying these carnivorous plants, which they always kill because they don't know how to water them and they don't have the right substrate for them, which I've definitely done. But paracarnivory can also exist in other ways. A lot of times it comes up in like sort of symbiotic ways. For example, there are plants that are called myrmecophiles. And myrmecophiles are plants that love myrmecos, or ants. And they like them so much that they have evolved with these ant species to essentially be a living nest. And it's very beneficial for the plant because those ants will kill pretty much any organism that lands on it, whether it's trying to feed on it or not. And typically, 9 times out of 10, it's something that's going to be deleterious to the plant, so it works out. And organisms that have adapted to this have found their own unique ways to just not be hurt or something like that. Or they don't, they die. So the ants kill the organism, right? So they kill it on the plant. But not only is the organism going to be wasting away on the surface of the plant, the ants are going to be eating the organism. They're going to be eating that insect or arthropod or maybe even a small mammal, and all of their bodily processes are going to beneficially affect the plant as well. And that makes sense, right? Because as you may or may not be aware, we exhale CO2 and we inhale a lot of things, but we process oxygen specifically. And that's useful for the plant because all these small little ant bodies are constantly inhaling. Well, they're not really inhaling like we do. They have a very different structure for breathing than we do. They don't have lungs in the same way. They're spiracles, but they're respirating, I should say. They're respirating and they're excreting all kinds of things, including frass or insect poop. And they're doing that in their tunnels, and they're doing that around the plant, and they let the carcass fall next to the roots, and then the plant will eventually benefit from that nutritionally. But it will also benefit from the CO2 respiration as well, directly in what are called domatia, or houses for the ants, which they live inside of. In some cases, plants have what are called extrafloral nectaries. So it's exactly what you think. They're places where organisms can get nectar that aren't in the flower. Some plants have no flowers that bear nectar, and they just only have these extrafloral nectaries. And some have both. But it's also helpful for the ants too, right? Because the plant produces this sugar content for the ants or whatever organism eats it, and then the organism hangs around and, maybe leaves a nice, really rich, nitrogenous gift or present, in the form of frass. And, that nutrient exchange there is kind of what happens in a symbiotic relationship. And it's when something is paracarnivorous, it's an organism that maybe benefits in that similar sort of direct way, but it doesn't do anything. It doesn't adapt it in a way to like directly kill the organism. Maybe it attracts other organisms that kill it for it. So there's sort of this indirect relationship going on. It's not one to. It's not plant to, plant to pest. It's sort of plant to predator to pest or something like that, or a parasite or something. So it's kind of a neat thing. Interesting to note that carnivory is something that has, I think, if I'm remembering correctly, at least, carnivory is as a trait is evolved in some plants and then sort of goes away and then can come back later on in the same sort of lineage. So you have this sort of like, I guess you could say like development, these sort of development periods that happen. And for those who don't know, like, for example, for Venus flytraps, they are carnivorous, and part of that is because they need to get that nitrogen, and they live in bogs and swamps, traditionally anyways, and they aren't very nitrogen dense, so they have to get their nitrogen through other means, or at least they can't access the nitrogen, I should say. Yeah, ants domesticate aphids to get honeydew from them, says Chip the Ripper 01. Yeah, exactly. That's trophobiosis, but that's sort of similar, right? Although I wouldn't say that it's necessarily paracarnivorous for the plant, it is a relevant thing that ants will do on plants with other bugs. But instead of killing them, they feed them and herd them, which is not great for the plant, but is great for the ants. Which was reminding me of another sort of thing with regards to that, but now I've blanked. What was it? With ants and that sort of symbiosis. Oh, right. So, like, for example, with mycorrhizae, so fungi that have a relationship with plants, a lot of times, if it's a mutualistic relationship, the fungus provides something for the plant, and the plant provides something for the fungus. And a lot of times it's an exchange of like inorganic phosphate or phosphorus rather, or it's an exchange of like phosphorus for sugars or other nutrients for sugars or something like that, like trehalose or hexose or something like that. And in a way, that's kind of the same thing as these myrmecophytes, there's myrmecophytes, these ant plants, right? Because they provide the domatia, they provide this sweet nectar, and the ants gravitate towards that because of the nectar and the food source, and then they produce frass, and they produce exoskeletons, and they collect their waste elsewhere out of the plant. They don't keep their nest dirty necessarily, but all of this sort of stuff combines, and they're sort of exchanging things, but for what is waste for them is treasure for the plant. So you have this sort of mutualistic relationship. Similarly, mycorrhizae is that way. The mycorrhizae aren't necessarily producing what would be waste for it, but they are signaling and exchanging these nutrients with the plant, like sugars and phosphorus, for other compounds or for like water or that sort of a thing. So you have this sort of mutualistic relationship. I was reading in a research report recently where somebody was talking about how in a particular context that I don't remember, which is really important to the report anyways, but they said something to the effect of symbiotic relationships like the ones they were talking about are common to occur in places where there's an ecosystem where there's a lot of organisms, and I don't know how they define a lot, But also when it's difficult for, like, there's a high carrying capacity, but it's really difficult to, like, survive, I guess. I mean, where is it not difficult to survive, and what ecosystem are we talking about? And I might be misremembering some of the details now that I think about it. But the point being is that the symbiosis, essentially, it's sort of poetic. At least my interpretation was poetic, because the symbiosis occurs primarily because that organism is, or those organisms need to survive. And so in order to do so, they kind of band together. And it's not like a conscious effort, It's, I mean, they might have an instinct or something like that. They don't, like, necessarily cognate in the same way that humans would, which is an important distinction to make, but I think it's actually even more impressive than us trying to figure it out, because we already have the mental faculties in order to do so. And this sort of symbiosis is mostly born through strife. You have the same sort of thing with mycorrhizae. You have these ant plants in the same sort of fashion. You have these paracarnivorous plants that, like, benefit from organisms kind of, like, doing their own thing, but by doing their own thing associated with the plant, you have an advantage for the plant, and the other organism is loosely benefited or not benefited directly at all. So you have, for example, parasitism on one end, where one subject or one population benefits and the other population or subject is not benefited, and in fact is detrimentally affected by it. And on the other hand, you've got a mutualistic or symbiotic relationship where both parties are benefited. And you have this sliding scale, and on one, you have a mutualistic sort of, light side of the force, and you have, like, a parasitic or a commensalic in the middle, sort of gray or dark side of the force. But of course, that's a human sort of ordering and structuring. There's no organism out there that's, like, on the good side or on the bad side because they're aware of it. It just happens to work out that way over millions of years of evolution. Anyways, I really appreciate you guys waving. Calbud, Budstead, Evil Nor, VQ Max. Am I supposed to actually read people's names like that? Your names are sometimes difficult. Shipmaster 420, Shipmaster. Were you on a ship? Shipmaster? Is that why your name is Shipmaster? Chip and Ripper. Yeah, root exudates, right. So root exudates are one way in which plants can communicate with microbes, like mycorrhizal fungi and diazotrophs, which are like the rhizobia in the soil that produce nitrogen, or they cleave molecular nitrogen N2 from the atmosphere in the soil. So when you have, like, bean plants or plants that are able to fix nitrogen or make those relationships with nitrogenous organisms, that's how you're doing that. That's how they're doing that. You have those microbes, they produce nod factors or nodulation factors, and those factors communicate with the plant, essentially. I'm simplifying it a little bit, but they communicate with the plant, and the plant will create these tumors, or through some means of, like, sometimes genetic or hormonal exchange, or not exchange, but development, these sort of nodulation, these nodules are developed, and they envelop the diazotroph or the rhizobia, and then they kind of just stay there and live out their lives fixing nitrogen. So it works out for them in that sort of same way. They get protection, they get a constant food source, and the plant gets a really important nutrient. My last name is Shipley. I am master of the Shipleys. Okay, not the ships. Alright, well, that's a totally appropriate reason to name. I'm not going to name shame you for that or anything like that. Concierge Nursing Recovery joined. Darcy Dabbs joined. I appreciate that. I'm hoping that these live feeds are becoming a little bit more entertaining. I like to ramble, but I at least want to ramble with a topic that's, like, kind of concise and structured a little bit, I suppose. I wish I was master of ships. Well, maybe just master of one ship. I would definitely take one ship. Then, if it was between 0 ships and 1 ship, I would take 1 ship. I'm totally with you there. Mr. Feelsmore joined. Thank you. Thank you. Why am I thanking people? Well, I do appreciate it, I guess. I guess that was a little bit of heart to heart there. I wasn't even thinking about that. But yeah, so I was looking back on my post because I knew that I had posted about that Wageningen University device before, and it was all the way in April, and at the time of this recording it's October 23rd, 2018. Very informing, keep it up, says Cal Bud. Well, I appreciate that. I'm glad. Very glad to hear that, for sure. I wish I was able to find the post that I had on paracarnivory, but I don't seem to be able to find it, unfortunately. A lot of stuff on the termite gut microbiome. I guess a lot of that research was going on around the same time that I made that post. Yeah. Does anyone have any questions or things about what I've been talking about or what the other live feed was about? This is sort of a continuation of that live feed, I suppose. Yeah. Maybe I have something here. Well, that's interesting. Yeah, I remember this post. So, I guess another thing about the symbiosis with mycorrhizae, for example, is that so you have on the individual level, you have the colonizing fungus or group of microbes. It's probably not just one organism. There's probably multiple organisms interacting at different seasons, different times. There's extirpation where some of the populations are they go extinct in a local area and then they get repopulated, and that sort of a thing happens. But now I'm getting questions, so I'll shut up about that. Mr. Feelsmore says, if a fungus gnat eats a spore of Beauveria bassiana, is that enough to infect it or whatever? More spores on contact is better. Maybe it can happen. It can definitely happen with one spore, but it is way, way more likely to happen with multiple spores. And not only are they going to be ingested, but also they're going to coat their whole body. They'll get into their spiracles, which is how they breathe. They'll go in through every orifice if they can, but they'll attach to the cuticle, the skin, essentially, of the plant, of the organism, of the insect, and they will sort of penetrate through the cuticle that way. I came in too late, not exactly sure what you're talking about. Yeah, I'm not surprised, because I just kept rambling. Glad you're able to share. I have DE on top of my pots and have been bottom watering, and every few days apply the spores to that water. I know they die quickly in water. Actually, diatomaceous earth is not an effective control for fungus gnats. You might find that it works for you, perhaps, and that's good. I'm not trying to tell somebody not to do something if it works for them, but at the same time there are research reports that sort of go into detail about how it doesn't necessarily work, at least under laboratory conditions. But if you have a small population, maybe it helps a little bit. I actually think Beauveria bassiana is superior to Bacillus thuringiensis, but that's just me. That's been my experience. As for what I was talking about, Calbud, I was talking about a lot of things. I was talking a little bit about a post that I made in April of the Wageningen University phenotyping device, the thing that was the subject of the last live video that I'm merging onto this live video. So I was talking a little bit about that post where they go into greater detail, which I thought was amazing, and I was ecstatic about it. And then I was starting to talk about the subject of symbiosis and paracarnivory in plants, which I thought was kind of cool, but totally unrelated to any of that nonsense. So that's what that is. Yeah, so, and then I have this post that I was talking about in March regarding how, so you have individuals who are, individual plants that will create this relationship with microbes, but sometimes those relationships are shared between different organisms, different individuals of the same species in a population, or different species entirely, and you get a community effect where like a mycorrhizal organism shares the nutrients and water and other sorts of things from one tree, like an oak tree, to a pine tree, maybe even to other sorts of plants. And so you have that sort of mycological network people like to often talk about, but not in the detail that makes it, to me, truly fascinating. Just that, like, everything's connected, man, and it's actually way more than that, and it's actually kind of cool to talk about it in detail, at least for me, as I'm obviously talking about it. And I'm glad that you guys are giving advice to each other in the comments about Beauveria bassiana and Bacillus thuringiensis and that sort of a thing, because I think it's really helpful to establish this sort of community acknowledgement of what works and what doesn't work. And that's the only way we'll get better, is if we all kind of pool our resources and not... I mean, competition is a thing, and competition can be effective in some cases to, like, make people strive for excellence. But in my opinion, I don't think it's very helpful to just keep all the secrets locked away and to only give them to a few people, because at the end of the day, we need not to sound like a revolutionary, but we do need a bit of a revolution. I am often talked towards by people way older than me in the industry about how we need a lot more younger people in ecology or in agriculture, but it's like, yeah, we do. And you know how you start that? You make farming a little bit more attractive. Maybe start paying your guys a little bit better. Maybe stop beating your workers or relying on, like, immigrants who don't, they don't get treated very well necessarily. Like, I mean, that's been my experience. I definitely know a lot of people who are disenfranchised. They're either immigrants or they're poor, and they just don't have a lot of choices, and they're mistreated for it, and like, it doesn't really reflect very well on agriculture in general. You know what I'm saying? So, not everyone's like that, but that's been my experience. Not personally, but I've observed it myself. Yeah, the soil food web, says Darcy Dabbs. Is sand as infective as diatomaceous earth? Well, neither of those are infective. They don't infect anything. But as a barrier. No, not terribly. If you keep it really dry, though, it would have an effect. And if you're bottom watering, I could see it working essentially the same as keeping the top layers of your soil dry. I could see that, because the fungus gnats, they don't want to oviposit onto moist soil. Moist substrate is like the number one reason why people get fungus gnats, or at least get them in the large quantities that they do typically. Yeah, definitely. So maybe try that out. I don't have any like peer-reviewed academic information on the subject, so I can't speak to that. But I could see how that would work with the right sort of setup potentially. But then I might even say the same thing about diatomaceous earth, and I know that doesn't work from laboratory research, so maybe not. But try it out. Yeah, overwatering, and you're saying overfeeding on nitrogen. I suppose, but I think that's way more ancillary to the moisture aspect of the problem. But yeah, good question though. Good question. Watering control works in combination well. Yeah, right. So yeah, definitely gotta be good about the irrigation. I knew this group of people who were overwatering all of their young plants, and this is in like a hydroponic greenhouse setting. They were using rock wool, and it's like, obviously, like, you don't even have to know plants to like kind of intuit that like the younger plants are going to need less water, or you will overwater them and kill them. Like, unless you've never ever like raised a plant and you've never overwatered, which is like the number one thing gardeners even do. So like, unless you've never had that happen to you, and that's probably going to happen if you've never like tried to raise plants, then I really don't understand how people don't get it. Maybe they're just like, unenthusiastic about their situation, or they don't care. It's probably a nicer way to, or a less nice way to put it, but definitely like being very prudent with how you water your plants is important. And propagated plants are going to need different water requirements than fully adult and mature plants. And if it's a plant that keeps growing bigger and bigger, obviously the water requirements is necessarily going to change and possibly go up in a related fashion. How do fungus gnats even get into your environment other than flying or clinging onto you? Right, so, and then we see soil or amendments. I think they live in the living soil and feed off of it, so their unborn are in it, or the compost you add. Yeah, so those are all good examples of how any real or any soil-borne organism, whether it's a pathogen or a pest, can come into your grow. So, if you go out and just collect soil, you don't know what's in it. So that's one way that you could have an organism get into your soil and get into your grow that way. And even if you do buy packaged soil, they can still be in it sometimes if they're not properly sterilized or stored properly. If there's a hole in the bag, then something can get in there, obviously. If it gets wet, like all kinds of things can happen. Absolutely. Fungus gnats are going to fly into your area, and despite their name, and I misspoke earlier, I do have a video on fungus gnats coming out, but it comes out on the 30th of October because it's super spooky. But yeah, it's not out yet. If you're a Patreon subscriber at the cultivation support level, then you would have access to it. But since nobody has access to it until the 30th. And fungus gnats do not, what I was going with this is that fungus gnats do not just eat fungi. They do eat the living soil. They will eat fungi, no doubt about it, but they'll also eat plant tissue. No problem with that either. They will eat totally not fungally infected, no rot plant tissue. So they're really plant gnats, not fungus gnats necessarily. Yeah, super spooky, right? Outside soil, they'll be attracted to the moisture. They'll fly around for a suitable place to live. Humidity can also be an attracting factor, certainly. So there's a ton of ways that organisms can get into your cultivation area, be it outdoor or indoor. And of course, if you're outdoor, you're exposed to all of the elements, not just the sunlight and the nice air, but also like aerial pollutants, and not that I'm trying to like rag on anyone who grows outdoors. I'm just saying that you are literally exposed to the outside, and that encompasses all of the outside, and the fungus gnats are part of the outside, so that's where the relationship starts and stops. Do they not want to eat cannabis because of the terps? Nope, that is not a problem for them at all. Seems the terps would be too much for them. Nope, not at all. Especially not in the root system. Not a whole lot of terpene production going on in the root system. Maybe if somebody developed some mutant cannabis that produce a bunch of the terpenes in the roots, that would be possible. Do you think most likely it came from your Fox Farm soil? Maybe, but not necessarily because the Fox Farm soil was tainted on site or anything. Maybe that's possible too. But it's possible that the storage was bad, or there was a hole in the. It's hard to really speculate without knowing all of the context, which even you probably don't have. So it's hard to say. Maybe it's possible. It definitely wouldn't be the first time that would happen to somebody. So it's definitely a realistic possibility. Yeah. Actually, speaking about whether or not fungus gnats would be disincentivized to eat on cannabis plants because of their terpenes and other constituents, kind of by definition, what makes a pest is it's the inefficacy of the plant's natural defenses to defend it. Because if the plant, it's like a chicken and the egg kind of thing. If the plant were able to, affect the pest, then it wouldn't be a pest. It wouldn't be problematic for the organism, or for the plant, right? So that's kind of what makes a pest is that it's really good at living on the plant and killing it or maiming it. It's impossible to know the whole route your soil took before it got to you. Yeah, exactly. It's impossible to know that route, but if they're professional, they would do things like, depending on the kind of soil, they would like sterilize it, or they would source it from the right place, or they would create it in a way that does not expose it to those sorts of things. But in transit, in transportation, it could be totally fine on site, and then, midway through its travel, become inundated with something. So it's hard to say. But what is the best way to eliminate plant gnats? It depends on what you're doing and how you're growing. But like I was saying, one of the most important things you can do is make their environment not very conducive to living. So fungus gnats spend the majority of their time as larvae, and that's where they do the most damage, besides also vectoring things like Botrytis cinerea, and vectoring Fusarium and Pythium and Verticillium and other sorts of fungal pathogens, either as larvae or as adults. So that's problematic. The best thing that you can do is you can make the first few inches of the soil mostly uninhabitable. That's where the sand idea came from before, because somebody would maybe try to put sand on there to keep the first few inches really dry, and then feed water from the bottom, so that you essentially make that area that the fungus gnat would normally inhabit not inhabit. And that's one thing you can do, and I think that's the most important thing you can do, sort of a cultural control from an IPM standpoint. You don't have to spray anything necessarily, although you can also add to your system, your anti-fungus gnat program. You can apply like Beauveria bassiana or another biocontrol agent that might kill it outright. You can drench it into the substrate that you're growing in. It might make sense to do that. It might not make sense to do that. It depends on what your resources are and how big your grow is and a lot of other factors. I would never tell somebody who was like only growing like 5 or 6 plants that like you should maybe do that if you don't have the money to just drop on like Botanigard, for example. But if you do, that might be helpful. But I think it's most helpful to just kind of deny the area to the fungus gnat larvae, because if they can't exist in that environment very well, then they're not going to reproduce well at all, and they're not even going to seek that location out to put their eggs in, stopping the cycle in general. That also includes your property too. So a lot of people, they focus on their indoor area or their cultivation space, but they don't even like consider the like inches or centimeters outside of their cultivation area, where like right next to them they might be having this big like water soaked like little grassy knoll or whatever. Maybe there's a ton of fungus gnats in there, or Fusarium or Pythium or something like that. Can you do that during flower? Can you drench with Beauveria bassiana? Absolutely. Absolutely. I mean, as long as it's allowed in your state and all that sort of thing, which is another important subject to talk about with regards to like whether you can or can't do something. Different provinces and states have different like laws about that sort of a thing. So you should check your laws and talk to your agricultural commissioner. I got beneficial bugs. The Beauveria bassiana, I bottom water applied several inches of sand, put up sticky traps. It's a battle, but I am confident I will win. I'm confident you will win too. Yeah, beneficial bugs, Beauveria bassiana, and also a cultural control. So you've got 2 biocontrols and 1 cultural control. That's really good. Keep good hygiene too. Like don't let like, it's hard because if you've got a living soil system, you kind of want this sort of like microbially rich decomposition happening in your soil potentially, but like again, you maybe run the risk of making a habitable zone for fungus gnats. So it's kind of like, I don't want to say that you can't do that, but it is one of the realities of that sort of a system. Oops, not sand. DE might add additional sand to it though. Yeah, in the video that's coming out about Bradisia species fungus gnats, or dark-winged fungus gnats, those are the ones that pretty much everyone come across. Those there's like 2 research reports by a man named Raymond A. Cloyd, who I've met several times. He's an entomologist that I look up to. He's a very cool guy, and he knows quite a bit about many different pest organisms. And he talks about, in those 2 research reports, he's an author to them, and he talks about how diatomaceous earth does not work for the fungus gnats in laboratory conditions. So you can check those out in the description of that video, because I always want to back up what I say with academic literature. What about beneficial nematodes for fungus gnats? I heard they eat the larvae. Yeah, they can. Like Steinernerma, I think I mentioned actually Steinernerma felchiae, and 2 other species, I believe, are used. The problem with fungus gnats and nematodes, in my opinion, is that you have to constantly reapply them, and they can be kind of expensive. But if you're a commercial grow and you have the resources for it, I say do it, absolutely. But if you don't, then it's not a very attractive outlook. Sitting water is my biggest issue with it right now, but trying to put 6 drops of spores per gallon to the feeding trays. 6 drops of spores, but how many spores in a drop? That's the question. You want to get more spores on contact if you can. The higher the spore density with Beauveria bassiana and other sorts of entomopathogenic fungi, the better, because you increase the chances of infection. Just like how being in a place where there's a high volume of, mosquitoes with malaria increases your chances of contracting malaria. Same sort of a thing. It recommends 1 drop per gallon. Okay, but at what rate? Right? They're not that expensive compared to all these other organic sprays on the market. You can get 10 million of them for $30. Right, but you have to use all of them at the same time. You can't use half of them, or unless you have the special packaging, I don't really know. But a lot of the times, you can buy 10 million or whatever, and that's a little bit overkill depending on how big your operation is. But it's mostly that you have to continually repetitively use them. And in some places, and some products are more expensive than others. So it depends on where you're getting them from and whether or not the quality control is up to snuff. 1/10 of a syringe treats 25 yards or so. Oh, okay, well then excellent. Then, if that's in the label, then you should follow the label. And if it's not working, double the label. Not with everything, but with Beauveria bassiana, it's more spores on contact is good. Beauveria bassiana is not restricted in Canada. Yeah, exactly. Is it possible for a plant to be immune to a pest? If so, how might that work? And then Calbed says, you can get them in smaller packs. Yeah, okay, so that's what I'm saying, is if you have the right packaging set up, then that works out fine. And if you can, if you only need a little bit at a time and the packaging accentuates that, then that's excellent. But every grow is a little bit different. I'm glad that the nematodes work for you. I don't want to sound like I'm poo-pooing it. It's just that I guess it's just been my personal experience. But I do know that they work really well, and EPNs are something that are very much in vogue right now in biocontrols, and they have been for a long time. As for the question for immunity to a pest, is that a salt gun behind me right here? Yeah, that's exactly what that is. That's right. Also, if you can't see, there's a salt gun, there's the sword, and then there's the this butterfly net, which I thought looked kind of cool. Oh, but back to your immunity question. So, yeah, it is possible for a plant to be immune to a pest or a pathogen. There's different kinds of levels of immunity. For example, you have immunity, which means not affected and not infected at all, basically, which is self-explanatory. You've also got you've got something called type 0 immunity, and what type 0 immunity is it's when an organism literally can't infect or attack this plant because of some usually really fundamental thing, like the bark is too thick or it's just not the right kind of food. So that's, if it's not the kind of host that it normally eats, like if it's a specialist, then it's type 0 immune, for example. So like, a predator bug that doesn't go after spider mites, well, the spider mites are type 0 immune to that predator, as an example. That's not how they really look at it for biocontrol agents, but you see where I'm coming from? So you've got tolerance, and then you've got resistance, and then you've got immunity. At the tolerance level, it means the plant can sort of tolerate the organism without too much damage, and that's sort of vague. And then resistance means that they can mostly kind of beat it back if it's a pathogen, for example, or a pest. Something about the phytochemistry in its leaves or something can affect it in a way that would, like the terpenes, might make it repel it, or it might poison it, possibly, depending on the organism. And then immunity is like outright immunity. It does not affect it at all. There's a lot of ways that can happen. And natural selection kind of helps out with that, and so does breeding and artificial selection. I haven't tried it yet. I'm just asking you. I bought some to take care of my thrips, and I hear that they take care of fungus gnats as well. Yeah, try it out. Definitely. Yeah, hopefully that question was that a useful enough answer for you for the immunity question. It's a really complex topic, to be honest, and we're learning new things about plant physiology and the interaction between different symbioses and plant pest pathogen or predator biocontrol agent sort of trophic levels and interactions like that. It's really cool. It's really fascinating. It's basically just pathology. It's phytopathology, right? So that's what that is. Yeah. The medicine man joined. Hey there. But yeah, I think that that's kind of what I wanted to talk about, and I think that it's about time. So if you are just getting here like the medicine man is, and if you have a question, you should type it now. But I'm probably going to go now, and I'm going to add this video and the last video to my YouTube channel, so you can get all of that information there, and subscribe if you want to get this information and you just don't happen to be at the live. I will upload my live feeds, pretty much all of them, to my YouTube channel as Zenthanol Live, so you can look that up, Zenthanol, and yeah, so we have another person who's joining too. I feel so bad leaving when people have just joined. Thanks, bro. Have a good night. Yes, you too, all of you guys. Yeah, again, if you didn't come across this video in time, you can find it on my YouTube channel, which is also in my bio, and you can subscribe to it if you would like, because that's how you get, that's how you are signaled that I have a video that came out. Anyways, and this will be available for 24 hours, so listen to it. If you have any questions or comments, just send them to me, and I will talk to you all later. Have a good one.